Thanks, everyone. So I'm Nicola Hemmings. I'm a researcher at the University of Sheffield. My work broadly tries to understand what the drivers of variation in reproductive success are in wild animal populations. And I really do focus primarily on the causes of hatching failure in birds. I work on a lot of different bird species, but as I'm sure many of you will be aware, over the last 3 years or so, I've been running a project focusing on curlew hatching failure and really working with a lot of you here and some other people who aren't here to try and understand what's going on with curlews at the egg stage. And I put this big slide of text on to start with because I really can't stress enough how much this project absolutely wouldn't have been possible without the input of all of the people working with curlew on the ground who have been working with us, helping us collate data, sending us samples to analyse over those years. So I just really wanted to basically make sure I started by saying a massive thank you for that. Whilst you're looking for your name or your organisation's name on the list, and if I'm very sorry for any omissions, and if there are any, please let me know so that I can fix that for future. I just wanted to give a little bit of history of the project for those of you who are maybe unfamiliar or just as a reminder. So it was back in 2021, and I'd already been working on hatching failure in a range of other species, including black-tailed godwits. And through those connections that I had with people through these other investigations, some people had asked me if I would analyse a very small batch of curlew eggs that had failed to hatch. It was just 12 eggs that were sent to me. And those eggs that year, I noticed that more than half of them had what appeared to be really kind of thin, crumbly shells. And obviously, it was a very small sample, but it just triggered this thought in my mind that actually, I wonder if this could potentially be an issue. And then I realised actually, I don't know much about hatching failure rates in curlew, and I couldn't find much consistent information about that either. So at the same time, the people I'd been talking to who'd sent these eggs, they had ideas about problems that curlew might be facing. So, for example, because recruitment is quite low, the expectation is that the age structure of the population is probably changing and shifting towards older individuals. And so there was some concern that this might affect fertility. So, for example, if you have lots of older males, then you might have lower fertilisation rates. At the same time, I knew from work that I was familiar with in domestic poultry that if you've got lots of older females, you might start to get problems with egg quality as well. So this really kind of sparked my interest too. So that year, with the support of the Curlew Recovery Partnership, I applied for funding from the Royal Society to conduct a 3-year study gathering data and samples from breeding curlew across the UK and Ireland to try and first of all, understand what kind of rates of hatching failure were like, and then also to try and see if we could get at some of these drivers. He would be really annoyed with me if he knew I'd put this massive photo of him up on the screen. But I just also wanted to kind of make sure right at the start I acknowledged the role of Dr. Jane Thompson in this. So the funding that I got from the Royal Society, most of it actually went to employing Jamie, and I'm sure many of you here will be familiar with Jamie from correspondence over the last few years. He did an absolutely fantastic job of helping me coordinate this project, and he also led on a lot of the data collection, some of the lab work as well, and data analysis too. So unfortunately, the funding that we have for this project did come to an end last November. So really sadly for me, but good for Jamie, he's moved on to another job now in the Environmental Agency, but I'm still in very close contact with him and he's still helping kind of finalise some aspects of this work. So our key aims in this project were, first of all, to determine the rates of hatching failure in UK and Irish Curlew. And also we're kind of interested to see if these varied, maybe across regions, habitat types, see what the variation was like between years of the project as well. As Mary said before, we're really looking at hatching failure aside from predation. So we know predation is a big problem, but we're actually thinking about those eggs that remain intact in the nest beyond the end of incubation and that fail to hatch for reasons that we don't really know. And that's what we were really focusing on here. We also wanted to establish the relative importance of fertilisation failure versus embryo mortality for causing curlew hatching failure. And also look at embryo mortality in more depth. So to try and understand when embryos are dying during the developmental period and why this might be. And then finally, because of that idea about potential eggshell or egg quality in general, we were also interested to assess sort of egg traits that are associated with hatching outcomes. So across the 3 breeding seasons, 2022 to 2024, thanks to all the efforts of everyone involved, we compiled data on egg outcomes from a total of 1,428 eggs from 381 wild incubated clutches. So I'm going to use the words wild incubated to refer to those eggs that were left in nest and kind of incubated by the parents. And then also on 348 eggs from 97 artificially incubated clutches. So these are eggs that were taken for head starting. And we also got lots of data when we, as well as the hatching outcomes, we got lots of data on kind of things like locality, so that we could look at differences and different spatial scales across the 3 years. As well as the data and hatching outcomes, we also received 224 unhatched eggs that to our lab that we could analyse to look at what actually happened inside those eggs. 78 of these were recorded as abandoned, and the rest were kind of unknown. I'll come back to that abandoned point in a second. But all of these eggs that we received into the lab, the egg lab, we examined microscopically to determine whether they'd been fertilised or not, and also to stage any embryos that we found inside the eggs. So, what did we find? So what I'm going to tell you now is kind of broad data across the 3 years, just to give you a kind of overview. So we haven't got enough time today to really dig into all the nitty-gritty, but I'm very happy to answer any questions about kind of the more specific stuff at another time. So we found hatching rates varied from 13 to 19% across, hatching failure rates varied from 13 to 90% across the years. So 13 to 90% of eggs failed hatch. That's for the wild incubated eggs. And remember, this is not including eggs that were predated. So this is just the eggs that were kind of left in the nest. And for head started eggs, the rate was 14%. So key thing here is hatching failure is quite considerable in Curlew. However, we did, a couple of years ago, we did a cross-species meta-analysis of hatching failure rates. And it was the first one that had ever included threatened species. So there'd been quite a lot of these kind of analyses historically that pitched hatching failure rates across species at about 10%. We, in our meta-analysis that included threatened species for the first time, the rates were up to about 17% across species. So actually, Curlew do seem to be sitting in what we would expect for a threatened species. So it's not good, but it's also not wildly kind of higher than what we would expect. So for wild eggs, the exact proportion of hatching failure really depends on whether we include those abandoned eggs in our calculations or not. And ideally, we wouldn't include those abandoned eggs because we already know that the reason they failed is because They were abandoned, right? And if we're interested in the other drivers of hatching failure, so things like egg quality, maybe something to do with the nest environment, maybe environmental contaminants and things like that, then that's kind of beside the point. Those eggs wouldn't have hatched anyway because they were abandoned. However, there is a bit of complication with that assumption. Last year, Jamie and I published some work which highlighted how abandoned eggs can cause a bit of a problem for the accurate estimation of hatching failure rates. And we used the Curlew data as an illustrative kind of case study for this. So 5.5% of failed Curlew eggs in our data set were recorded as abandoned, but we have evidence both from the eggs that we received, but also from a kind of systematic review of the literature on this topic, that abandonment can often be incorrectly or inconsistently recorded. And I'll give you some examples of how we know that. So first of all, in some cases, eggs are recorded as abandoned, but we know that other eggs in the clutch hatched. So in that case, the parent has obviously stayed in the nest and incubated to the end of incubation period, and that abandonment has been recorded afterwards. But the likelihood is that egg failed before that the parent left the nest, because obviously the other chicks hatched. And we also found in 20% of clutches that were recorded as abandoned, we found really big variation in the developmental stage at which the embryos inside the eggs were at. In some cases of 10 to 11 days of development difference. So obviously abandonment can't have been the reason that all of those eggs failed. Some of those eggs, at least, embryos died before that clutch was abandoned. So we kind of have this spectrum of uncertainty, which means the true value of hatching failure likely kind of lies somewhere between that 13%, which excludes all abandoned eggs, and about 90% which include them. And I think that's just something, that level of uncertainty is something that we just need to kind of think about and incorporate into how we understand hatching failure in the future. It's really hard to get accurate information on abandonment. It requires, to be super accurate, you need to basically be having really high nest visit frequency, and I just know that that's not feasible for a lot of the people that are working on Curlew. There's just a kind of person power issue. But being aware of the uncertainty and being sure of our records of abandonment would be kind of useful in the future. When we look across regions, we see quite a lot of variation. So you can see in this in the text at the top, quite big variation in hatching failure rates across different areas. Some of this variation might be due to differences in sample sizes that we've received from these different places, but also it's important to remember that lower hatching failure rates in our data set could just mean much higher predation rates, because obviously if the egg is predated first, then you're never going to know whether or not it would have gone on to successfully hatch or fail. So some of the areas that look like they got quite low hatching failure rates might be suffering from really quite high predation rates, and that could explain some of that variation as well. You can see in these maps that we did break down the data into even kind of finer resolution. I'm not going to go into that in any detail now, apart from just to say that really there's not that much to say. We found very little evidence of any association between hatching failure rates and latitude and longitude. So things are fairly consistent across the areas that we looked at. Hatching failure rates also exhibited some variation across breeding habitat types. Again, sample sizes are really variable across these different habitat types, so we haven't done a kind of formal analysis of this, but as you can see, we've got some very high rates of hatching failure in eggs from arable and military breeding habitats. Arable is probably mostly predation. And lowest in those from moorland habitats. I'm happy to talk about any of the details of this at another point. Again, just in the interest of time, I won't go into too much detail there, especially given that we're going to have a huge amount of data. In terms of egg fertility, of the eggs that we received to examine, 88% of those were kind of definitely confirmed to be fertilised. The other eggs, however, they weren't confirmed to be unfertilised. They were simply too degraded for us to be sure. And we found very similar. I haven't got this up on the slide, but we found a very similar situation with the abandoned eggs that we received as well. Most of these were fertilised as well. So I think, we're very confident that fertilisation failure isn't a major problem at the moment, at least for these curlew populations. So I think we don't need to be worried about things like, sperm quality, copulation behaviour and things like that. These eggs are being fertilised. That's not the problem. However, the problem is, for hatching failure in curlew, it certainly is embryo mortality. So I'm just going to talk you through these plots just so that you can kind of understand this data in a little bit more detail. So these are all the eggs. These plots show you all data from all the eggs that failed because of embryo mortality across the 3 years. And so in these bars here, we've got head-started eggs. And in these bars here, we've got the wild incubated eggs. And what we've done here is we've broken down embryo mortality into 3 distinct phases of embryo development. And these are important phases because they are really different phases in terms of what the embryo is actually, the processes that the embryo is going through. So phase 1, which is in the pink part of the bar, is the very early phases of the kind of formation of the body plan. So very early basic body plan formation for the embryo. This happens in just the first few days of incubation. And then in the middle, in this kind of orangey red colour, we've got a period of very rapid cell division and differentiation of body structures. And that takes up kind of the bulk of the middle of the incubation period. And then in the last few days of development, we have this period of rapid growth. So there's no new body structures being formed. There's no differentiation happening. Everything's in place. They are just growing. And what you can see across our data is particularly striking for the wild eggs is that the majority, it's about 70% of embryos that die are dying in this late stage of development. And it struck me yesterday, I think it was Stuart who was saying something about how, how it's so awesome to see those legs when you're ringing. Well, it's really not so awesome to pull those long, fully formed, looking ready to run legs out of an egg that you've got sat on the side of your lab. And actually, this is something that was actually quite upsetting for my group. So many of these chicks look like they should have just hatched. And so I think this is something that is really worth thinking about. There's not some kind of fundamental congenital defects that's stopping these embryos from developing properly. They're getting so close, and then something's just going wrong at the last minute. And so, that's something that I think would be really worth thinking about in the future. For the head-started eggs, there is a little bit of a different situation. So a larger proportion of those eggs are failing early in development. I've got some ideas why this might be. So I've worked with other species head starting programs, and I know that this is a common situation. And in other species, it's thought that this third couple of days of development can actually be really sensitive. And so disruption in terms of the movement of eggs, disruption to incubation in those early stages could cause problems. And I know one head starting program for a different species that actually opt to leave eggs in the nest for 1 or 2 days before taking them for artificial incubation, and that does lead to improved hatching success. So that, you know, that also might be something worth thinking about in terms of head starting kind of procedures. Okay, so we've kind of established that embryo mortality is the primary driver. We've established that we've got high rates of late stage mortality, and that's kind of the kind of headline stuff that we've figured out. But we have been running I want to kind of take just these last bit of time just to tell you about a couple of additional projects that we've been running to try and get at what might be causing this embryo mortality. And these data are a little bit more preliminary, but I think we've kind of done enough to be able to share this with you now and at least make some kind of inferences about what might be going on. So I mentioned at the start about this question of eggshell thickness. And one of the things that we've been doing is looking at how eggshells vary between hatched and unhatched eggs. And we've been focusing on those late stage eggs because we know that's where most of the mortality happens. So you can see from these data here, we looked at both eggshell, effective eggshell thickness, which is this section. This is a cross section of the eggshell. This is the bit that doesn't change throughout development. This is like kind of the hard outer shell. And this bit here, which I'll show you in a second, this is the mammillary layer. This bit does change, and I'll explain why in a second. But the key thing to say is, contrary to what we expected, there's actually little difference in this effective eggshell thickness across all regions of the egg, the blunt equator and the pointed end of the egg. Between hatched and unhatched eggs. So, okay, doesn't look like there's much difference there. However, when we look at this mammillary layer, which is actually the part of the egg where the embryo draws down its calcium for its skeleton, we do see a difference, but it is in the opposite direction to what we thought. So actually, mammillary layer of the failed eggs is thicker than it is for our hatched eggs. And we don't think this is just because those hatched tits have used more, because we're really comparing the embryos that died right in the last stages of development when they're basically kind of fully formed already. So this is interesting. One of the key things that is known, again, from the poultry literature is that poor quality eggshells tend to have mammillary layers which are disorganized, with these mammillary knobs that you can see here are often large and irregular. And this can happen because of issues with the female reproductive system. It can happen because of issues of female age. So this is something that we found quite striking, and we definitely would be really interested in looking into more detail. But certainly that eggshell quality could be an issue. And then finally, very quickly, I'd like to tell you about a project that we ran last year, which is a bit different, but I think you'll find quite interesting. I certainly found this very interesting, the data that we got from this. So a master's student of mine was using the curlew eggs to look at something that we've been looking at across some other species as well, which is the presence of microplastics in eggs, of whether that's linked to egg viability. We've got a stream of work on environmental contamination in the lab, and this is kind of part of it. So the master student, Daisy Whittle, she basically took curlew eggs. She did this whole process of digesting and filtering the egg contents to extract microplastics, and then she looked at what was inside those eggs. She found a range of different microplastics in unhatched curlew eggs. In every unhatched curlew egg she looked at 41 eggs from the 2024 breeding season. She found, this is a small sample size, but I think it's quite convincing, because each of these kind of pairs of dots you can see are eggs from the same clutch. So she found that there was a real kind of relationship to the concentration of microplastics in eggs from the same clutch. So it looks like exposure to microplastics is quite consistent, which kind of makes sense. And then finally, again, she only had a small data set, but she looked at whether there was any association between microplastic concentration in eggs and the hatching success rate of the clutch those eggs came from. Now, the important thing to note here, there is some weak evidence that there might be an association, but it's mostly driven by this single clutch, which had 0 hatching success and a particularly high rate of microplastics. And if you take that one out, you can see you kind of lose that effect there. So something I'd be really interested in is getting more data on this to see if this holds. And also the other thing that we don't know, and I'm actually not really sure how we'd find out, but it's something I've been thinking about, is what happens in the clutches that we don't know about that have got 100% success rate? Are they relatively clean? You know, is there some kind of threshold level at which contaminations start to impact eggs? And so I think that's really interesting in itself. And I just think as well, although these data are very preliminary, I think the fact that we find microplastics across all these eggs really just should really alert us to the fact that environmental contaminants could be a future problem for these birds and wildlife in general. So in summary, we've kind of established hatching failure rates across these populations. They're high, but not beyond what we would expect. Fertilisation failure is rare, and hatching failure is mostly due to late-stage embryo mortality. And just these issues of eggshell quality and environmental contaminants could be a kind of concern for the future. Okay, I'm going to finish there and just say thank you again to everyone who helped with this project. I really, really appreciate it.